for the questions.
Okay.
Okay, we can also hear you, please.
So good afternoon. You will have to switch to the next slide more frequently. I will present you a paper that was also selected as an exceptional achievement by the Slovenian research agency, and it deals with the protein complexes that we found in oyster mushrooms, Pleurotus, that could be used as new bioinsecticides. Please, can you go to the next slide? This is just the distribution of these small aegeralysins proteins that can be found especially in different fungi and in bacteria. And today I'm going to focus on these Pleurotus striatus aegeralysins. Next slide, please. Pleurotus mushrooms are edible mushrooms, and they are known to produce two important protein families. Proteins from the protein families that act together. One of those are aegeralysins proteins, shown here in yellow, and their names, the abbreviated names in the next slides will end with A. And another proteins are MACPF proteins, no? So proteins with membrane attack complex domain that is lipic. So these two type of proteins produced by oyster mushrooms are known to form transmembrane pores. These pores are practically a bicomponent composed of approximately 26 molecules of an aegerolysin, a yellow protein here, only A, and 13 molecules of the MACPF protein partner that is responsible for this penetration through the membrane. Next slide, please. So what is the role of the aegerolysin proteins inside this pore? Aegeralysins are known to be proteins which specifically bind to membrane lipids, and they especially bind to two kind of lipids that belong to the class of sphingolipids. Sphingomyelin, that is that is the main sphingolipid of vertebrates, and ceramide phosphoethanolamine, that is the main sphingolipid present in invertebrates, especially in insects, in some protozoans, and in some gram-negative bacteria. And at the right, you can see the association constants of these proteins with the membranes containing these sphingolipids. You can see that aegeralysins interact more than 100 times stronger with these insect-specific sphingolipid. So the presence of these sphingolipid, which is specific to insects and not found in the cells of vertebrates, and the ability of aegeralysins to bind to these lipids and to form transmembrane pores in concert with their MACPF protein partners, led us to perform some insecticidal tests in collaboration with Agricultural Institute of Slovenia. Can you go further, please? And here are some of the tested insect pests. And as you can see at the right, the only beetles that were, the only insects that were susceptible to our proteins that died after the treatment were Colorado potato beetle and western corn rootworm, that are also one of the economically most important pests of corn and potatoes. Next slide, please. Here are the survival curves after treatment of Colorado potato beetle with our protein complexes. The larvae and adults were fed with potato leaves soaked in our protein complexes, and after the first ingestion of these food, they stopped to eat, and they died of starvation. You can see below in the petri dishes that treated larvae were much smaller from the untreated, and the LD50 was quite low. Next slide, please. And the same effects were observed also with western corn rootworm. Next slide, please. So to conclude, I would like to say that proteinaceous bioinsecticides are not new in agriculture. Since 1960s, some bacterial proteins are used, and these are proteins from bacteria Bacillus thuringiensis. There are 300 of these proteins approximately, and their mechanism of action is by binding to protein receptors in the insect midgut. The problem is that many insects have developed resistance to these proteins, and one of the mechanisms of the resistance is well, modification of the protein receptor. Well, one of the possibilities that our aegeralysins MACPF complexes could have some future to be used there as bioinsecticides is their ability to bind not to a protein receptor but to a lipid receptor. So in the constitutive molecule that is building also biological membrane, and this is lipids are less prone to modifications and mutations. So perhaps the chances of developing resistance from the insects could be lowered. These results are also covered by an international patent application. And if you can go further, please. I would like to thank, of course, the Slovenian Research Agency for financing us, and special thanks to the Knowledge Transfer Office of the University of Ljubljana, who helped us a lot. And on the right, there are the people that contributed to this work from our group. Thank you.
Okay, thank you for this nice presentation. I will have a question myself. In the morning sessions, we heard that one of the important factors is regulation, which influences the speed of commercializations of such very important environmentally friendly technologies. And what is the case in this field? So is there already some regulatory established on the level of European Union?
I don't know. Well, our complexes are now being tested by one of the companies that have expertise in this plant protection. The results are quite good, and you know, we are basic researchers, and we need the help of another partners. I could not answer this question. I'm just an ordinary biochemist.
Okay. Thank you for the presentation again. We will move to the next one.